Hello everyone, my name is Darby Tassel and today I will be presenting a study on the effects of nicotine on activity rhythms of zebra finches, or Teniopygia guttata. This study was performed with Wes Pyatt and Dr. Noah Ashley of the WKU Biology Department. So, first, some background. So the Ashley lab has been studying the circadian rhythms of birds in Utqiagvik, Alaska. These birds primarily are the Lapland longspurs and the snow buntings of the area. And these birds experience 2 months of 24 hours of daylight. This coincides with their mating season, so it's a very important time for them. Therefore, we want to see if their circadian rhythms during this time affect their fitness. So if we want to manipulate their circadian rhythms in the wild, we need to figure out if the ways that we are manipulating their circadian rhythms are safe and effective. That's where lab studies come in. So in the lab, we study these effects using zebra finches. Zebra finches are a good test species because they are similar in anatomy to the snow buntings and Lapland longspurs. They're all passerine birds, and so they're a good model species. So this study, as I said earlier, focuses on nicotine. Nicotine is a stimulant in humans. It keeps them awake for longer. And so we hypothesized that this may do the same thing in zebra finches. We hypothesized that it would promote activity and that there would be a dose-response effect, so higher concentrations would promote more activity than lower concentrations. So, methods and materials. So, obviously the animals that we used were adult zebra finches. We used both male and female finches. These were obtained from the aviary at Western Kentucky University. So to use the nicotine, I used nicotine hydrogen tartrate salts, which was dissolved at various concentrations in distilled water. The control dosages were just the distilled water, and they were injected with a hypodermic needle. To monitor their activity levels, we used computer software. This computer software is called Limelight. The birds were placed in separate cages with adequate food and water, and as well as a perch. And so also the lights were kept on for 24 hours to simulate the conditions in Utqiagvik. So this software monitors the activity of an animal. It was built for mice, but it also works for birds. It basically works for anything that you want to move around in an enclosed area. And so it would graph 10 minutes of movement for 24 hours. Basically, it would take the data from 10-minute intervals, and it would repeat collecting data for 10-minute intervals for 24 hours. So let's move on to the results. So this is a graph of the data from birds that were injected with 0.12 milligrams per milliliter of nicotine. So as you can see, there is not a lot of effect on their activity rhythms. Like the peaks happen in essentially the same areas, and the valleys also seem to happen in essentially the same areas. If anything, the control distance has a higher level of activity, but it's not really clear if these are outliers or if this is actually an effect of the nicotine. So next, we get to higher dosages. So, the 0.12 didn't work, but, maybe that was it was too low of a dosage. However, 0.16 milligrams per milliliter of nicotine also does not have a huge effect. On this one, the colors are switched, but as you can kind of see, there's a lot of data that's sort of just near the floor, near the x-axis here, but still there's just not really a lot of variation between the two. So then we finally get to our highest dosage, which is 0.2 milligrams per milliliter. And there's a little bit more data on this one, but even so, there still isn't an extremely significant effect of nicotine on the data. The peaks and valleys happen in essentially the same areas. On the second graph, the blue graph, there is a little bit of difference in the valley of the control, but the peaks of the experimental distance are lower, so it's not really clear if this is like really disruption of sleep or if it's just, again, some outliers because each of the birds would have like 144 data points. So even though it may seem like there's a lot that are up there, it's really not a hugely significant number. So analysis. So as you said earlier, 0.12 milligrams per milliliter doesn't really do a lot. There has been other data recorded for this experiment that does not really show anything significantly different than the data presented here. However, we also figured out that our dose response effect wasn't really showing up like we thought it might. So 0.16 milligrams per milliliter, it doesn't have the clearly defined peaks and valleys like the other data, but it also doesn't have that significant of a variation. Finally, 0.2 milligrams per milliliter didn't have that really disruption in activity peaks and valleys. However, it did seem to possibly affect the bird's behavior, albeit not really in sleep patterns, but it actually seemed to affect their health. So after injection, occasionally the birds would shake and vomit and stay in one place for a significant period of time. So there was a possibility actually that immediately after injection, nicotine could have the opposite effect in these birds. We don't have a lot of evidence for that yet, but it definitely was not promoting activity in the way that we expected. I feel like it's important to note all of the birds ended up okay. They all, whenever I would come back in the next morning, they would all be sprightly and flying around and terrified of me like they usually were. But anyway, next, so I think it's time to discuss some possible errors. So Limelight is not a very widely used software, and therefore it is not updated extremely often. So it has some issues with how it tracks activity. So, I mean, one thing is that it was built for mice. So, however, it is really the only software that can be used for this purpose. So, you know, but, and another important thing is it will create false readings. So it will monitor a lot of activity that may not have happened. So it's really important to look at the video artifacts, and sometimes these are not created depending on possible issues with the computer. Another thing that would occur sometimes is that data of 0 centimeters would be recorded when it was clear that activity had occurred and the computer just was not picking up on it. Of course, the 0 centimeter data, whenever that was consistent throughout a bird's day with the trial, the data would not be used because it just didn't show anything. Another thing is that because nicotine causes weight loss in humans due to appetite suppression, we had a thought that this might also occur in the zebra finches. However, there really isn't any substantive data to prove this. So body mass was recorded before and after during the 0.2 milligram studies, and during these, the birds, some of them did lose weight, but some of them gained quite a bit of weight proportionally. The birds only weigh about 15 grams, and sometimes they would gain a gram, which is kind of a lot for their body weight. And finally, as I said earlier, nicotine could have the opposite effect on zebra finches as it does in humans, because another stimulant that works in humans is caffeine, and this stimulant has the completely opposite effect in house sparrows. It causes lower activity levels. This was shown in previous studies performed by the lab. This study is not published yet, though. So our conclusions. One thing is that nicotine does not have the desired stimulating effect on zebra finches, and by extension, most likely other small birds that would really make it useful in the field. There's a possibility that it could have a depressant effect, but there's not really enough evidence to support this interpretation. And then finally, and most importantly, we cannot really advise the use of nicotine in the field because it does not disrupt sleep patterns or activity rhythms, and there is a distinct possibility that it could be hazardous to the bird's health, and that is not the kind of thing we want to be putting out in the ecosystem, especially one as fragile as the North Pole. So that being said, here are our acknowledgements and our contact information, and Thank you so much for watching, and